Hi everybody, this is Mrs. Kraus, and this is our video on the Unit 2 Part 3 notes on macromolecules. So within our cells, we have 6 elements that are found in very high concentrations: carbon, hydrogen, nitrogen, oxygen, and phosphorus, and sulfur. And you can remember those 6 elements with the acronym CHNOPS. You take the first letter from each element. We already know that water is extremely abundant in our cells, and water contains 2 of the elements in CHNOPS, hydrogen and oxygen. Okay. Most of the molecules we talk about today, in fact all of them, have a carbon base. Why do they have lots of carbons in them? Well, it's because carbon can actually form 4 covalent bonds with other atoms. That means it can share 4 electron pairs. For that reason, carbon is very good at building long chains. Now, the discipline of studying molecules that contain carbon is called organic chemistry. And we have 4 main molecules that we're going to look at today that are carbon containing and found in your cells. The first one is carbohydrates, which are also called sugars. The second is lipids, which are also called fats. The third is nucleic acids, one of which is DNA. And the fourth one is proteins. Okay. Now, I used the term macromolecules before. If we break down that word, macro means large, so these would be large molecules. Now, they're typically formed through a process called polymerization. Polymerization is when you take many small molecules, which are called monomers. For mono, think one or a single unit, and you take those single units and you connect them together in a long chain. That long chain is called a polymer. Poly meaning many, so it's many monomers joined together. Each of the 4 macromolecules, carbs, lipids, proteins, and nucleic acids, have monomers that join together to make polymers. So this just shows us the process of our little monomers being joined together to create a polymer, and that process is called polymerization. Okay. We could also call polymerization a dehydration or condensation reaction. We call it that because monomers are being joined together. So sometimes we call it dehydration synthesis. Synthesis means to make, so we're making a polymer. And in the process, releasing a molecule of water. That's why it's called dehydration. Dehydration means to lose water. So let's say I had 2 monomers here, the smiley faces, and they both have a little OH on the end. When these 2 monomers get joined together, we are going to lose the H from one and the OH from another. So we're losing water to build a larger molecule. The opposite process is called hydrolysis. Hydrolysis takes place when you add water in order to break a polymer into monomers. So it's the opposite of dehydration synthesis. Instead of making a larger molecule by losing water, we are breaking a larger molecule into smaller parts by adding water. Hydro means water, so we're adding water, and lysis means to break. Okay. Again, our 4 macromolecules are carbs, lipids, nucleic acids, and proteins. Let's get started by talking about carbohydrates. Carbohydrates are also called sugars. They are a source of short-term energy for both plants and animals, and they are also used in plant cell walls to provide structure. Carbohydrates always contain the elements carbon, hydrogen, and oxygen, always in the same ratio. For every 1 carbon you have in a carbohydrate, you have 2 hydrogens and 1 oxygen. For example, glucose is a monomer or single building block of a carbohydrate. It has the chemical formula C6H12O6. If we divided all those numbers by 6, we'd end up with 1 carbon, 2 hydrogens, 1 oxygen. So always the same ratio. Now, the monomer of a carbohydrate has the generic name monosaccharide. Mono meaning 1, saccharide meaning sugar. To remember that saccharide means sugar, I always think of saccharin, which is an artificial sweetener found in like diet sodas. So single sugar. Okay. Again, these are carbohydrate monomers. And some examples of monosaccharides are glucose and fructose. Fructose is a fruit sugar found in like berries and basically all fruit. And glucose is a sugar that's made during the process of photosynthesis in plants. Okay. A disaccharide, di meaning two, is when you take two monosaccharides and you join them together to form a two-sugar carbohydrate. An example is sucrose, which is table sugar. Sucrose is actually a glucose monomer and a fructose monomer stuck together. If you were to join many monosaccharides together in a long chain, you get a polysaccharide. Poly meaning many, saccharide meaning sugar. So this is the carbohydrate polymer. Remember, all of our macromolecules, carbs, lipids, proteins, and nucleic acids, have monomers and polymers. Some specific examples of polysaccharides are starch, which is used for storing energy in plants, glycogen, which is used for storing energy in animals, and cellulose, which is found in the cell walls of plants and gives them a really tough cell wall. Something I find interesting is glycogen is actually stored in your liver, and let's say you're exercising, that glycogen gets released from your liver into your blood to provide you with energy. If you are exercising pretty hard for more than 3 hours, you're going to deplete your source of liver glycogen, and you're going to have to start taking in Gatorade or those little goo packs or the little gummy bears that they use for people in marathons and triathlons and stuff because you've gotten rid of all of your sugar stores in your liver. Interesting. Okay. This is just showing you a starch molecule. Basically, a starch molecule is a bunch of glucose molecules joined together to form a long chain. That's all it is. Okay. Our second macromolecule are the lipids or fats. They are made of mostly carbon and hydrogen atoms, but there are a couple oxygen atoms as well. So it doesn't have the same ratio, the 1-to-1 ratio that carbs do. It has a lot more carbons and hydrogens than it does oxygens. Lipids are used for several purposes in cells. They're used for long-term energy storage, insulation, stay warm, and also they're found in cell membranes. Some examples of lipids are fats, oils, and waxes. Okay. Now, what I've drawn here is a typical fat molecule, which is the type of lipid. A typical fat molecule is made up of long chains of carbon surrounded by hydrogens. Those long chains of carbon surrounded by hydrogens are called fatty acids. There are 3 of them in a typical fat molecule. So we would have this same red molecule here and here. I just got too lazy to draw it. Your fatty acid chains are always carbons and hydrogens, and they are always nonpolar, which means they repel water. That's why when you put oil, which is a lipid, in water, they tend to separate from each other. They're not attracted to each other because the oil is nonpolar, the water is polar. Now within this fat molecule, there's also this blue part. That's glycerol. It does contain carbon, hydrogen, and oxygen, and for that reason, it is slightly polar. But the fatty acid chains are so long that overall this molecule is nonpolar and is not attracted to water. So in a fat molecule, we have 3 fatty acid chains and a glycerol. We often call fat molecules triglyceride. Tri for 3, glyceride, I believe, describes the 3 fatty acid chains. This is a simplified picture of a fat with the glycerol molecule over here containing carbon, hydrogen, and oxygen, and the 3 fatty acid chains here containing just carbon and hydrogen. Now, there are 2 different kinds of fats, saturated and unsaturated fats, that might be part of your diet. Saturated fats like butter are solid typically at room temperature, whereas unsaturated fats like olive oil are liquid at room temperature. What's the difference between the two in terms of their structure? Well, saturated fats contain fatty acid chains like this one shown in red that have no double bonds between carbon atoms. So you see only single bonds between carbon atoms. So that means they're only sharing one electron pair with each other. For that reason, the fatty acid chain is straight. If you were to put these fatty acid chains on top of each other, they would stack up really tightly packed. Because they're tightly packed, they're solid at room temperature, they're very dense, and in the human body, if you eat too much of that, that could result in some clogs in your blood vessels. You'd form some plaques within your arteries. Unsaturated fats have at least one or more carbon-carbon double bonds. So these two carbons are sharing not one but two electron pairs. That's what it means to be a double bond. That results in a kink or bend in the chain. So when you stack unsaturated fatty acids on top of one another, they're very loosely packed. They're less dense, which means they're liquid at room temperature. Typically, unsaturated fats in your diet do not cause plaques to form in your blood vessels. They don't lead to a clogged blood vessel, which could cause a heart attack or stroke. So they're healthier for you than saturated fatty acids. Okay. Next on our list are the nucleic acids. They are used to store genetic information and to transmit or send that genetic information in the form of a code. They contain carbon, hydrogen, and oxygen, just like carbs and lipids, but they also have nitrogen and phosphorus. The monomer or single building block of a nucleic acid is called a nucleotide, and if we join nucleotides in chains, we can create two types of polymers, DNA or RNA, and we'll look at the differences between those in just a second. So the monomer for a nucleic acid is a nucleotide, and the polymer is either DNA or RNA. Okay. So here we're showing you the structure of a nucleotide, which is the monomer for a nucleic acid. It consists of a nitrogenous base, which is shown right here as a little double ring structure. Nitrogenous just means it contains nitrogen. Then we have a 5-carbon sugar. I know that this pentagon is the 5-carbon sugar because it has 5 sides and 5 points. A 5-carbon sugar might also be called a pentose sugar. And then we've got a phosphate group, which contains the element phosphorus. DNA is 2 chains of nucleotides that are wrapped around each other to form a spiral staircase-like structure. Right. Now DNA, its full name is deoxyribonucleic acid. It is the master copy of an organism's genetic code. So it contains all the instructions needed for all the life processes that occur within that organism, and those instructions get passed down from parents to offspring. This is a picture of the DNA molecule. Notice it's 2 chains of nucleotides that are wound around each other. RNA, or ribonucleic acid, is used to make a copy of a short section of DNA, and that copy of the short section of DNA can be used to create proteins. Creating proteins is also called protein synthesis. This is RNA. The major difference that I see here between DNA and RNA is that RNA is just a single chain of nucleotides. Okay. And we're going to do a second video for the protein so we don't run out of time.